Thank you very much for such a warm welcome. Really, Ms. Superville, Professor Carrington, friends, colleagues, it really is a great pleasure for me to be here today and to welcome you to this awareness workshop, specifically aiming to increase our interactions and cooperation between ICGEB and Trinidad and Tobago. As you've all heard, our relationship goes back a long way now. Trinidad and Tobago has been a very valued member of ICGEB for many years, and I'm very pleased to see that we've had some excellent collaborations and some very successful programs have been run within the country. And I should say that the workshop that Professor Carrington mentioned that she ran on bioinformatics is really a model of the type of workshops that I would say ICGEB really wants to support and is very happy to see succeeding. It's this ability to bring together people from around the world and have a truly international workshop so that people can learn from each other, which is really at the core of what we aim to support. So as you'll hear in the next hour and a half or so is a general overview of ICGEB and what we're about. And then there will be more specific information about our different programs and basically some words of advice on the best ways to approach the ICGEB in terms of content, both for applications for meetings and courses, what we're looking for when we receive applications for fellowships, and of course, what we're looking for when we receive applications for grant grant funding. I should stress that in addition to those, we have a lot of activities related to technology transfer in a range of different sectors, and those programs can be particularly bespoke. And so if, when you've seen the presentation of what we do, if there's any interest in additional forms of cooperation or collaboration with the ICGEB, then I'd be more than happy to discuss those possibilities in greater depth. So really what I'm hoping for here is that we will see a broadening of the number of people within Trinidad and Tobago who know about what we do, and will develop an interest in expanding the collaboration with us. And finally, I should stress that our membership is now 66 countries located throughout the world. Many of those countries are in your geographic location, so Central America, South America, obviously also much further afield. And one of our major goals is also to make sure we can leverage that network of countries to promote international cooperation and collaboration. So don't feel that all of the activities that are going to be reported or described today have to be carried out in one of the ICGEB centers. We are very keen to promote scientific collaboration and cooperation among our membership. So I really do encourage researchers within Trinidad and Tobago, if they identify research teams in other ICGEB member countries with whom they really would find collaboration and cooperation to be a valuable asset to their own research activities, then to really use ICGEB to help support those sorts of interactions. So with that, I'll stop and let the people really start to disseminate the information, and I remain on hand for any questions or follow-up discussions at any point. So thank you very much, and I really look forward to a great workshop. And also I would add, please, if you're too shy to ask a question openly, use this chat, please, and we'll respond as the workshop proceeds to each of the questions that we get. So thank you.
Thank you, Dr. Banks, for your gracious welcome and remarks. So our next speaker will introduce the benefits of ICGEB's membership. Ms. Mariana Maculan is the Chief of External Relations at ICGEB, where she oversees the diplomatic political work of the organization to ensure there is fruitful relations with the member countries, along with the expansion of the membership base. She also works to nurture cooperation with partner organizations in the United Nations system and beyond. Further, she is the Secretary of the Board of Governors. Please welcome Ms. Mariana Maculan.
Thank you very much. Thank you for my short bio, and welcome everyone. So much has been already said, and we truly hope to see more of you in our laboratories, to meet more of Trinidadians in ICGEB. So I start by sharing the screen, and let me see. I hope you see my presentation. So this is the...
Yep, looks perfect.
Okay, so let's start by looking at our membership. So we have 66 member countries, and we are in an expansion mode. We are waiting the membership of 3 more countries that are in line and very advanced in the process. Our membership is very much spread all over the continents, and it's very Trinidad. In the Caribbean, basically we have Cuba and Trinidad and Tobago, but we also have many countries in Central and South America, and headquarters in Trieste. Then we have offices and laboratories in South Africa, Cape Town, and in India, in New Delhi. So we have the mandate. We are an international organization and a research center whose main purpose is to promote sustainable development. So it's completely aligned with the mandate of the United Nations. And how do we do this? Through scientific research, first pillar, training, second, and technology transfer. We will go through all of these 3 main pillars. And historically, we were created within the United Nations, the agency called UNIDO, Industrial Development Organization. And then in 1995, we have become a fully independent organization. So who's governing ICGEB? The Board of Governors. So it's made of all the member countries that get together once a year. This is the picture of the last time we were in person. And Professor Christine Carrington is representing Trinidad and Tobago at the board. The next session of the board will be in person, finally, after a few virtual meetings due to the pandemic. In May, we will be finally able to meet in person in Cape Town, South Africa, 10-11 May. So this is the main body, and it decides on the budget of the organization, on the main policies, strategies. Every 5 years, elect the Director General. And this body is advised and supported by the Council of Scientific Advisors, which is another important body of the ICGEB that is composed of 15 eminent scientists that supervise the science produced in our laboratories and make recommendations to keep it at the top of their game in each field of the life sciences. So, as I said, we have 3 main pillars. One is the scientific research that is carried out in our laboratories. We have 50 research groups that can be organized in 5 macro areas. One are the groups dealing with infectious diseases. The most numerous groups treat non-communicable diseases. Then we have the medical biotechnology. the industrial biotechnology, biofuels and industrial biotechnology. And then the fifth group is the plant biology and biotechnology. I give you just a very quick overview, and then through the workshop, my colleagues will go more deep in every one of these aspects. Then the second pillar is the capacity building, the training that we offer. So it's a very comprehensive scheme, a very comprehensive range of activities that we offer to the nationals of our member countries, and is composed of advanced education supported by fellowships for long and short term at the level of PhD students and postdocs. Then we offer competitive grants, also including early career return grants. And then we offer support for the organization of international meetings, courses, and workshops. So in 2021, we had in our laboratory 360 fellows. We are now finalizing the numbers for 2022, but it increased. So we are in expansion, not only as membership, but also as in the number of fellows that we are able to host in our laboratories. And this is the geographical spread from where the fellows come from. And exactly, we are doing this workshop in this moment in time because all the calls are open. So for PhD fellowships, the closing date for application is 31st of March. For postdoc fellowship, 31st of March, and then we have another call the 30th of September. My colleague Vittorio Venturi will go very much into the details of how to apply and how to apply successfully to our calls. I also wanted to add that when we get grants from specific donors, we open also other calls. Like these calls are every year the same date, and these are funded by our core budget. And whenever we get voluntary contributions on top of our core budget, we open calls. And normally they are like earmarked for specific activities, such as Italy gave us, Ministry of Foreign Affairs gave us a grant for women scientists from Africa. So we got a specific calls for that. And now we are implementing, the women are already in our laboratories, they are performing their fellowship. UN Office of South-South Cooperation gave us another grant for women from least developed countries to come to our laboratories, but South-South, so not Trieste. They went to India and to Cape Town. So we can receive voluntary contributions for specific projects that are agreed with the donor. Here is another program for which we opened a specific call funded by UN Tech Bank. So moving on to research grants. So we offer funding for scientific research. And this research has to be collaborative in nature. So it's to be formulated and implemented together with our senior scientists in ICGEB. And we have 65 ongoing projects at the moment from 35 countries. And I looked at the historical, and we were able to fund 2 projects in Trinidad and Tobago, but quite a long time ago. So we very much welcome applications. And also we support meetings and courses. We organized in 2023, 25 international meetings in 16 different countries. And also we give sponsorship for specific events. And the deadline to apply to this call is 28th of February. Again, Vittorio will go very much into the detail of this activity. I also want to mention that ICGEB believes in open science. So all of our international meetings, seminars, courses that we organized are recorded, they are all organized by subject, and they're freely available in our website, in iTunes and YouTube. So these are resources that we produce and everybody can take advantage of. The third pillar of our mandate is technology transfer. We work with the industries in member countries, and we transfer technology, especially for the production of biotherapeutics and diagnostics. So far, we have more than 100 agreements signed with industry in member states. And particularly our biotechnology development unit, BDU, develops technology for the production of biosimilars and then supports local biopharma manufacturing in member countries. So to improve the drug availability at a more affordable price. So most of the tech transfer are in this area. And in the map, you can see in green where we transfer the technology and the technology has reached the market. Whereas in blue is where we transfer the technology and they're doing the clinical trials. Of course, we don't do all this alone. We are very much partnering with a wide spectrum of partners from other universities and research centers to governments, to the United Nations and other multilateral organizations and foundations. For example, DDG mentioned the Bill and Melinda Gates Foundation is funding a big project. Again, that is including technology transfer of diagnostics on COVID disease, on COVID-19, and the technical assistance to empower this reference lab that receive the technology to implement it. So to summarize, being a member country of ICGEB, Trinidad and Tobago is very welcome to reap the benefit of this membership. And to summarize, this is access to a worldwide network of experts in biotechnology, in our own laboratories and in all the centers that we work with around the world. Then to access advanced education supported by fellowship for PhD and postdocs, organization of meetings, courses and workshops, competitive research grants, including early career return grants. Everything will be explained in much more detail soon. Tech transfers, access to state-of-the-art facilities. If you have a particular project and you identify that we hold a facility that you may need, you can apply for support to come to us, use the facility for a certain period of time to carry out your studies, and then go back with the results. And then provision of technical assistance and advisory service. With Trinidad and Tobago, we said we are your member since 2003. So the appointed governor, so let's say the more political voice of Trinidad and Tobago within the organization is Prof. Christine Carrington, whereas the liaison officer is Ms. Joanne Leon-Poy, and she's the main liaison between ICGEB and the scientific community, the one that will inform you about all our calls and will get the applications and make the first screening. So said that, I just terminate here. There is my email. I'm always available. If you have questions that may come later in time, please write to me. I'd be happy to give feedback. And I'm available for any question if you have any. Thank you.
Thank you, Ms. Maculan, for that insightful session where we learned about the three main pillars: scientific research, and the future speakers will talk about the 5 main areas: training and capacity building and technology transfer. We would now like to take a moment to open the floor for discussion. So what you can do is raise your hand and the IT help will unmute you and you can ask your questions, or you can type your questions in the chat. Okay.
maybe we can go on and then. Oh, there's some.
I see there's a hand raised.
Okay.
Hi, morning. My name is Lavan. I noticed Samuel said these opportunities were specific for PhD candidates and postdocs, but what about people with MScs in biotechnology?
Maybe Vittorio will answer.
Yeah. Hello, Levan. If I understand correctly, you're asking whether somebody doing a master's, currently doing a master's, can benefit from the ICGEB. Is that correct? Is that what you were asking?
Yes, that is correct. Well, after we have completed our master's in biotechnology.
Well, after you've completed your master's in biotechnology, if you want to pursue a career in research, the next step would be to do a PhD. So it would be, I would advise to apply for a PhD. If you're not sure that you want to do a PhD yet and you would like to do let's say a short training period, then my advice would be to contact the ICGEB PI of interest and initiate a conversation whether the PI has any possibility to host you in the lab for a period of a training period. Obviously, it would depend on the funding. You know, our pillar programs are for PhDs and postdocs, so we don't have any programs for masters. So I would advise to contact the PI and see if you can do a training period. If you're, on the other hand, convinced that you want to pursue a PhD, I think this is the best time to apply for a PhD fellowship. Okay.
Right. Thank you.
Thank you.
Okay, so we have another question from Vivek Maharaj. What are the membership fees? Not sure if I missed it.
Okay, well, I'll answer the two questions in the chat. With regards to membership fees, that's something that the government of Trinidad usually covers. So there are no membership fees to the applicants of the ICGEB programs. And regarding the recording, yes, we will share the recording of the session to all the registered participants following the session. You're welcome. Are there any other questions before we proceed to the next section of the program? Okay, so Chihiro, I'll hand it over to you.
All right, okay. Right, so if we are sure there are no more questions, I thank you all for your questions. We will now move on to guidelines for applying to the ICGEB programs. This segment will be presented by Dr. Vittorio Venturi, the scientific coordinator at the ICGEB. Dr. Venturi focused his PhD research on the regulation of ion transport processes of beneficial plant-associated bacteria, which promotes plant growth. He then moved as a postdoctoral fellow to the ICGEB Trieste, Italy, where he started investigating intercellular signaling among bacteria. He then went on to become group leader at ICGEB in 1998, continuing his studies on intercellular bacterial signaling. He is now interested in 1. How plant-associated bacteria undergo interspecies communication and interkingdom signaling with plants, and 2. Plant microbiomes and the development of microbial products for a more sustainable agriculture. I now present Dr. Venturi.
Let me share my screen. So can you see my presentation?
Yes.
Okay. Thank you, and thank you very much for the nice and warm introduction. And it's a pleasure for me to be here and to be discussing in an informal way what we offer as our pillar programs, as Mariana has already mentioned, and I would like to give you some tips and guidelines on what we're looking for and how to apply. Okay, so as Mariana mentioned, we have 3 pillar programs, that is the fellowships, the meetings and courses, and the grants. So I'll be following this I'll be following this order, and so I will start with the fellowships. So as a core program of ICGEB, every year we offer PhD fellowships and postdoctoral fellowships. And every year we have 2 deadlines for applying to these programs. One is the end of March, which is for the PhD fellowships. We only have 1 deadline. Okay, so you have to be very careful. There's only 1 application deadline every year, and that's the end of March. And you can see here, as Mariana already showed you, on the left here, the flyer that shows you the that actually publicizes this call with deadline on the 31st of March. When it comes to postdoctoral fellowships, we have 2 deadlines, and it's the same deadline as one of them is the same deadline as the PhD fellowship, which is the end of March. And also we have another deadline on the 30th of September. Okay. Now there are, I will now give you some information on the application procedure for PhD and postdoc fellowships. So they share basically the same application procedure. Now, if you're interested, the first thing you have to do before you apply is to contact an ICGEB group leader, an ICGEB PI. Now, I will mention later, we have, almost 50 PIs, or over 45 PIs at the 3 ICGEB components. So in Trieste here in Italy, then the component in New Delhi in India, and the component in Cape Town, South Africa. We have over 40 group leaders that do all kinds of research, right? Going from infectious diseases to communicable, to plant biology, to medical biotechnology, and also computational biology or bioinformatics. So if you're in the life sciences, we cover, of course, we don't cover everything, but we cover a large amount of topics in the life sciences. So if you're a PhD student, usually in the master's, you have developed a strong interest in a topic. If you're a postdoc, obviously you already done your PhD, so you have a much better idea what kind of research you want to do. So you look at our website very carefully, everything is there, all the PIs are there, what the research they do is there, obviously in brief, but also all the publications, the latest publications that they published are there. So it's very important you see our website and you make a decision on which one or 2 really fit your interest, your motivation. And then you contact the PI with a motivation letter. Now, let me say that this is a crucial step. Now, I myself am a PI, and I receive a lot of contacts, and I have to say many of them do not attract me, because many of them, maybe I receive a line saying, "Dear sir, I'm very interested in your research. I would like to apply for a PhD in your laboratory." You have to think that the PIs receive many of these kind of letters. So a letter like that will not attract my attention, because I don't know if the person who's writing is really interested in what I'm doing, is really motivated in my science, is really I'm a plant microbiologist, really has developed an interest in plant microbiology with a specific specific interest, a specific curiosity. So very, very often, I tend to reply to all, but most likely an email like that with a CV attached will not get very far. Okay, so my advice is that you really, if you're really passionate and convinced you want to do a PhD, is that you see where you want, and you have an idea where you want to do it. You write a motivation letter. It doesn't have to be too long. Again, being too long is not good. A good motivation letter, half a page, 3/4 of a page maximum, that you make it clear why you are interested to do a PhD or a postdoc, and that in the particular group of the group leader that you're contacting. And then hopefully that will work, and the group leader will reply, and a contact is made. So once a contact is made, maybe very often the group leader will ask you, let's meet via Zoom, or let's have a phone call. and then start talking to each other, and then together, driven by the applicant mostly, you prepare a research proposal. I mean, when I say research proposal, don't get scared. We're not looking for, like a grant application or a long 10-page proposal. It can be, research proposals can be a couple of pages, with a small introduction, hypothesis, and how you intend to do the work in order to prove or disprove the hypothesis. As I say, it has to be driven by the applicant, but you will receive feedback and support from the group leader, because if there is then a good feeling between the applicant and the group leader, it is also in the best interest of the group leader that the project is well written, cohesive, with a good scientific question, because then all this package will then be will undergo selection. So then obviously also the group leader will give you feedback and will help you, will guide you through the process. Okay, and this is also a moment where the group leader, and as I said already, the group leader and the applicant get to know each other. So it can also happen that along the talking by mail or Zoom, then maybe either the group leader or the applicant realize that maybe the interest of the applicant is not really in the field as much as the applicant would have liked, and maybe then the applicant can go to a more related field that he or she was looking for. Okay, so this is by far the most important step. I hope I've been clear that you have to really make an effort and engage with your passion and curiosity with the group leader. Okay. After this, actually, it's quite linear. It's quite the application procedure is quite fast now. Eligibility, Mariana mentioned we are a family of 66 member countries, growing, of course, which we're happy to say. The applicants of our pillar programs, so our programs funded by our core budget, so the money that comes from our membership fees and also mainly from the Italian government, is that you have to be a national of an ICGEB member state. So in this case of Trinidad and Tobago, if you're a national in Trinidad and Tobago, you can apply. If you are studying in Trinidad and Tobago, or you're doing a PhD in Trinidad and Tobago, and you're not a national of Trinidad and Tobago and you want to apply, but you're a national of the other 65 member countries, it's also okay to apply. Okay. We do ask for an English proficiency certificate, in order to make sure that, because everything, obviously, ICGEB is international, the working and teaching language is English. So we just want to make sure that your English is up to a standard. It doesn't have to be very, very good, but up to a standard that at least allows you to start activities. In the case of Trinidad and Tobago, obviously, this is not a problem. As I said, the closing date for applications is the end of March for PhDs and postdocs, and is the 30th of September for postdocs. Okay, so all the applications are online. Okay, so as I said, you have to, and here are the files that you'll be asked to upload in our system, and the CV, the motivation letter. Once the contact has been established with the PI, the group leader. So I say PI when I mean principal investigator. So PI and group leader is a synonym, is the same thing. So the group leader will provide an acceptance letter for you to upload. So he or she or group leader will say, I'm very happy to host this applicant in my laboratory in order to carry out this particular research. So that we know that the group leader is actually willing to host and has looked at the application and is happy to host you in the lab. An English language certificate. We need three referees, just the contacts, who will then send a letter by email to the fellowship's ICGEB email. If you're a postdoc, you can upload your PhD certificate and a document. Okay. Now, the PhDs usually last 3 years, but now 3+1. So, in exceptional circumstances, it can be finished in 3 years, but this is really exceptional. In most cases, the fourth, it takes 4 years or 3 years plus so many months to a maximum of 4 years. Okay. The postdocs, usually lasts 2 years. On exceptional cases, if the work is going very well and the proficiency is very good, then a 3rd year can be solicited to ICGEB for funding. You know, and it's not always the case, but it has happened that a 3rd year is also funded so that the postdoc can continue. So 3+1 for the PhD, with the 4th year usually taking place, and 2 plus possibly 1 in exceptional cases for the postdocs. Now, so these are the long term, so the long PhDs, the full PhD in ICGEB components, and the full postdoc, long-term postdoc in ICGEB components. Now, what is very important, and what also we are very active, are short-term fellowships. So we fund scientists who are doing their PhD in their home country, or or in another country. If you're in Trinidad and Tobago national and you are doing a PhD somewhere else, so our nationals from member countries that are registered for a PhD or a postdoc can come for a short period. This can vary. Usually, we like to say a minimum of 3 months, to a maximum, let's say, of 9 months, if they have a specific reason to come to the ICGEB components. Let me give you an example. You're doing a PhD in the plant sciences, and you would like to learn genome editing, CRISPR-Cas genetic engineering. We have this very well established in New Delhi, and you want to go spend 6 months there to learn how to do genome editing because you cannot do it in your home country. This is a perfect scenario where again the procedure is the same. You contact the PI. I would like to come to your group in order to learn genome editing. Again, you write a small project, obviously 6 months, very focused, providing a specific reasons why you want to spend those 6 months in the laboratory, and then you apply for that 6-month period. Can be 3, can be 9, depending on what you do. So you must have a specific reason to visit and spend time, a short time in a laboratory in ICGEB. But also, if you're a postdoc and you would like to spend a short time. Now, as a scientist, I can say that 3 months to 9 months is a very short time. Unfortunately, we all know that research needs time and passion and patience. So these kind of visits and projects have to be very well focused. There has to be a very specific reason. Okay. A third. We also fund these short-term visits. We call them smart fellowships. If it can very well happen that what you're looking for, a specific technique or learning how to do using a mouse, using a specific model, or doing some computational biology you cannot do in your own country, or you cannot do it at ICGEB because we don't have that, that particular expertise or facility that you need, we don't have it. Then and another laboratory in an ICGEB member country has, then you can apply for that laboratory. Or let me give you an example. The ICGEB 3 components do not offer a particular technique or that particular piece of equipment, but that laboratory in Brazil that I know has it. Then you apply for this short-term fellowship called SMART, not to come to ICGEB, but to go to laboratory in Brazil. Okay. Again, selections these are competitive programs. So the people who will review them are very have done so for many years and a lot of experience, most of them, and they will see very clearly if the project is well written, if there is exactly the reason that you want to go for a laboratory is justified. If it's not, due to the quite high competition, your application will not be granted. Okay, the short-term fellowships have 2 deadlines. They also have, like the postdoc, the end of March deadline and the end of September deadline. So all the short-term fellowships. And the application procedure is also online. It's just the same. So the application procedure is very similar. What differs is what you're doing, whether a full PhD, full postdoc, or short-term in ICGEB components, or short-term in a laboratory in one of our member countries. Okay. let me just finish this first part of fellowships. Well, almost finished by saying our fellowships cover all costs. So you have no, there's no, if you're doing a PhD, there's no registration cost to the university. You know, we as an institution, we cannot award PhDs. So we have agreements with universities, you know, in India and in Italy and South Africa. We have agreements with local and also international universities. All the registration fees are covered. You will receive a stipend for your living cost, which allows you to live in a comfortable way. You know, you can get all you need in either South Africa, India, or Italy, and also your travel to the laboratory, and once you finish back home is also covered together with and also you will receive a medical insurance as well. So basically, if you're awarded a short-term or any kind of fellowship in ICGEB, you don't have to worry about any costs. Okay. Now, as Mariana said, you know, what I've talked about, these are pillar programs that are launched every year and are covered by core funds from ICGEB. We also have extra programs. So my advice is always keep an eye on our website, because you know, our current DG, together with the management like Mariana and other offices here, are extremely good and active in looking for other donors, so we can make our programs, we can enrich our programs. So we're basically looking for more funding to have more programs, you know, for women scientists, or you know, or for LDC countries, or just more funding to have more specific programs. So visit our website. There could be, apart from the pillar programs, extra programs that you can qualify for and apply for. Okay, and these will have may have some slight variation to the application procedure, to the deadline, or to some. There might be some slight variation to the application procedure, but everything will be explained in our website. Okay. So that is the fellowships. I hope I've been clear. If not, we can have a question and answer session, and also, as Lawrence mentioned, you can contact us here in ICGEB and we'll be happy to answer any questions. Now I would like to move to meetings and courses. Meeting and courses, we have one call a year for funding, and that is always the call is launched, you know, early, and the deadline for the application is the end of February. So we're very close to the application. Okay. There are different categories of meetings and courses that we fund or sponsor. Briefly, I will go through them. You know, one of them is an ICGEB workshop. You know, so somebody in Trinidad and Tobago, you know, a professor or a researcher in an institute would like to organize a workshop. Usually what we look is for some co-sponsoring, co-funding. You know, this can be, for example, providing the seminar room, or providing maybe some of the coffee breaks, or some local transport. But usually, if we've got some co-sponsorship, it will make the application a little bit stronger. Sometimes we fund also events without co-sponsoring, but some you know, some co-sponsoring by the local organizing institute is I consider it important. Usually workshops are between 50 to 120 participants on a focused topic. You know, usually a topic that is actually at the moment important, and it will follow the classical way of workshops. So we will have oral presentations by invited speakers, and also by registered speakers as well. Some oral talks will be selected from the registered participants. Excuse me, registered participants. Some will be selected also to give oral presentation, and there will be poster sessions. This is, you know, the classical setup of workshop. The maximum contribution that you can apply for is 25,000 euros. Okay, so okay. Another type of event that we fund is the ICGEB course, right? Obviously, this is a course is for students. Like a workshop is geared towards anybody who can attend. You know, we always encourage PhDs and postdocs to attend. I'll come to how we would like to see the money spent. I'll come to it in a second. Whereas the ICGEB course is really focused completely to young scientists. So a course is really a teaching event, okay? And in most cases, it's theoretical and practical. We have had only theoretical courses, okay? Obviously, only practical courses really do not exist. Practical courses always have a theoretical component, okay? The participants are a smaller number, 25 to 50. And obviously here we're looking for the local organizing institute or university or to provide, it needs to provide facilities, equipment and staff. And here the maximum contribution is 15,000 euros. We like these very much. We like all of these, but ICGEB courses, we like them very much. And we also see more and more regionality. So an ICGEB course, for example, in Trinidad and Tobago can look at the whole Central American region or part of South America as well. We also fund what we call Future of Science. These are much, workshops tend to be 3 to 5 days. Courses usually are a week, sometimes even 2 weeks. ICGEB Future of Science events are very short, 1 day or 2 days, and it involves a very few high-profile leaders. Let me say, if there's something very important for Trinidad and Tobago, a scientific topic which is of particular relevance to Trinidad and Tobago at this particular moment, it's particularly important that we bring some really top scientists, leaders in a particular topic to Trinidad and Tobago for a specific reason, so that the scientific community is exposed to these high-profile scientists in that particular topic. Then you can apply for this event. So bringing these high-profile people, having a 1 day or maximum really 2 day event in order to promote this particular topic or to inform the scientists of this particular topic, this is what you can apply for. Okay, so I would not advise you that you apply for this for something that has a topic that is very well grounded for many years. In that case, my advice is to go for a workshop. And then we sponsor events, usually these are meetings that take place maybe regularly in our member countries. For example, annual society meetings. Okay, so we'd be very happy to sponsor these with for example, with 5,000 euros. And really, what we're looking for here, that one of our scientists is also, if one of our scientists can be invited to attend this, let's say, national meetings or even an international event that Trinidad and Tobago organizes, has already got quite a lot of funding, and would like to have an additional sponsor for ICGEB, so we can give ICGEB some visibility of our programs, and also then you can use some of that money to also bring some students to the event. In that case, we would like to see that one of our group leaders fits very well the topic of this event. We would like very much that he or she is invited to the event, so he or she can come, and then also it'd be an excellent way to do networking with ICGEB. Okay. All the application is online. You will find online several things will be asked from you. Obviously, they're shown here: the organizing institute, the scientific organizer, and I want to stop here. You can have co-organizers. So you can have, and the co-organizer can also be from abroad if you want. Very often we have one organizers. Sometimes we have 2, sometimes we have 3 or 4. Okay. We need to have information about the venue, where you want to hold it. A list of potential invited speakers. Again, this can be an indicative list. I mean, you don't have to contact them beforehand before you apply, because the application for this year, this deadline for end of February, it's for the events that will take place in 2024. Okay, so you will need to put also a tentative date here when you will host it in 2024. A list of invited speakers. Again, it's not necessary you contact them beforehand, but at least that we want to see that you've thought about the list of speakers, so we can see that it actually fits very well with the kind of topic that you want to hold, the topic of the event that you want to hold. A local staff, we would like to see who will be the local administrative organizing staff. Obviously, the subject area, and it will be asked what is the subject area, which justification of the subject area, also how you will select the participants. Usually, we can give advice here. We would like to see that the participants can be PhDs or postdocs, and they are actually undergoing research which is relevant to the subject area of the event. If we see that, participants don't have the particular interest in the subject area, that will not go very far. The budget, we want to see a breakdown of the budget. And again, here it's very important. My advice is that the budget that you ask for, let's say, if it's 20,000 or 25,000 for a workshop, that a certain amount is obviously for the speakers. A certain amount can be for logistics, but we would like to see that a certain number of young PhDs and postdocs attend the course, and they are fully covered in terms of local costs. So local travel and lodging and lodging and food. So my suggestion is put at least 10 participants from ICGEB member countries will receive a scholarship where they will get local costs. So usually international travel is not granted. Sometimes there can be a small contribution to local travel, but usually it's not. So be very careful. In the budget, we want to see that young participants from our member countries, a certain number, as many as possible, are covered. So if you put a budget that you will use all the costs for the lecturers or for the invited speakers, flights, and for maybe a welcome party, that will not go very far. We need to see young people being supported. Okay. And also we would like to see other sponsors, if possible. An event, unfortunately, we all know that events cost. So if you have other sponsors from companies or maybe national agencies, foundations, donors, even if you're going to contact them, it will be please indicate. You have to indicate that here, so we see that you're active in bringing, getting the funds necessary to have a nice event. Okay. So that's what I'm going to say for the meetings and courses. Finally, I will talk about the grants. For the grants, we also have a call a year, and the call is, this is the flyer for this year, and the deadline, as you can see here, is the 30th of April of this year is the deadline. And again, just like the meetings and courses, the deadline is 30th of April. The selection procedure will then take place, and projects will start on the 1st of January of 2024. Okay, so you're applying at this deadline to then, if successful, start your activities in 2024. The eligibility in this case is a little different. You don't have to be a national of the ICGEB member country, but you have to work in an ICGEB member country. So let me give you an example. If an American is working in a university or an institution in Trinidad and Tobago, he or she can apply. So in this case, it's not the national like fellowships, but it's where he or she is working. If he or she is working in our member countries, that's fine, because we want to. The idea of our CRP program, grants program, is to bring good science to our member countries. So the nationality is not important, is where he or she works is important. It is called a collaborative research program, but I want to immediately say that collaboration with ICGEB research group is welcome, but absolutely not mandatory. It must not be forced. I must bring in an ICGEB group leader in order for me to get this grant. Wrong. If the topic, if the research benefits, I'll talk now in a second, benefits to have the group leader because he's in the same topic and he's complementary to what I'm doing, then it's fine. If you don't have that, it doesn't matter. We call it collaborative because I will say in a second, collaboration for our program is fundamental, but it doesn't have to be with an ICGEB member states. I do want to say that if an ICGEB group leader fits very well to your collaborative thoughts, then go for it. It will be great, and then we encourage that. But if it doesn't fit, you must find another collaborator I'll mention from somewhere else. Again, the kind of topics we like that the topic that are addressed in the application are relevant to the country, right? Having said that, we fund good science. So if it's good fundamental science, that is also fantastic. If it's let's say a little bit more applied science, more translational science, then maybe it becomes a little more important that what the kind of translation that you're doing is relevant to your country. If it's top science, fundamental science, we do not care if the particular topic, the particular model or the particular biological mechanism that you want to investigate, the fundamental science really fits to some aspect of the country that becomes less relevant. So here we have the standard grants that apply. Then we have a special category which are called early career grants. So if you are a young scientist with a good CV and you spent a minimum 2 years abroad, then you can apply for a CRP grant and you'll say, I want to apply for an early career grant. Okay, and these type of grants, you should have spent minimum 2 years abroad, and you should not be older than 40 at the time when you apply. And you should have returned to, let's say, Trinidad and Tobago, no more than 2 years, right, prior to the submission of the application. So, for example, if you submit now, by the end of April 2023, you really should have come back, not before 30th of April 2021. So basically, looking for young people that have recently come back to Trinidad and Tobago, again of any nationality, and have a good CV and are not older than 40. In this case, if you fit this category, when you apply, you will tick the box. I'm an early career grant, and you will follow a different track in the. So we know that the selection team will know that these are early career grants. Okay, and each of our member country can, and I'll mention it in a second, can submit so many applications to ICGEB, and these are 3 standard grants and 2 early career. Okay, what are we looking for a successful project? I will mention it in a second, but it is also sad for me to say that we get a lot. Well, we're happy that we get a lot of good applications, and many of them very good. But the funds are, we can only award a certain number, as I will show you in a second. So the competition is very high. So if you intend to apply for a CRP, my advice to you is make it as perfect as possible. Okay, don't do it last minute. Don't do cut and paste with a previous application thinking I'm going to cut and paste. Really do your best because the competition is very high. So we're looking for, obviously, we want to see a clear hypothesis and address a specific question, of course, in the life sciences. Okay, we do only life sciences. Okay, sometimes you get applications which involve, for example, biomedical engineering. Already involves machines or something already, they don't qualify. So life sciences. Be realistic in what you're proposing, and also that it can be achieved in the proposed time. I've been involved in the selection for many years. Very often I come across applications which propose so many experiments. So I want to do, I want to sequence the genome, I want to do RNA-seq, I want to do proteomics, I want to make knockouts, and then I want to test it. And I know that work program is unrealistic, both in terms of time and in terms of funding. So don't think that, oh, the more I put, the more I include, the more I show I can do, the better it is. No, because then it will work against you. If it's not realistic, we're gonna immediately reject it. It's a very important point. I think people think they can impress the reader by saying they're doing so many things, but actually it can really work against you. Yeah, we want to see novelty. Obviously, that's you will hear that word. As a scientist, you hear that word all the time when you submit publication. We want to your results have to be novel. When you put a grant proposal, novelty. Of course, we know it's difficult to be very, very novel, but we want to see, even if it's something, let's say, that has been done maybe in another model system or something that you want to that has been in part done. We want to see that what you're going to do actually extends it or puts a novel twist to it. Confirmatory projects, so projects which maybe have been done in one organism can just be done in another, tend to be weak, tend to be rejected. Pure screening projects tend to be rejected. So the element of novelty, an element of mechanism usually is what we like to see. Okay, you will have in the application as a line, you will have a certain number of words. Be very concise. Okay, if you need less, write less. Okay, don't forget the selection committee has a lot of projects to read. So make life easy for them. Make them see that it's clear, it's concise, it's novel. Okay, and that will work for you. Okay, I've already covered this topic about I've already covered this issue about the relevance to the topic. Now there are two aspects which these kind of points that I've mentioned can be applicable to almost any grant that you apply. It doesn't have to be an ICGEB grant. It can be, most grants, these points actually are important. Okay. Now for ICGEB CRP grants, there are two aspects which are fundamental, that if you don't have, you will get rejected. Collaboration. We want to see that the laboratory in Trinidad and Tobago, the project has one or more collaborators which are complementary, which bring in a new technique or a new aspect, a new expertise, a new angle to the work, which is not present which is not of competence to the applicant. And ideally, this collaborator has to be international. Now, it's not enough to say, oh, I will find a collaborator in the US who will do proteomics with me or will do, so-and-so with me. No, that will not work. We want to see name, surname, and if he or she can provide a support letter saying, oh, I'm supporting the application of so-and-so, of Dr. So-and-so working in Trinidad and Tobago, and I will, the title of the project, and what I will bring to the project is this, this, and this, and this. So to see that the collaborator is very interested in the project and what she will bring to the project. That is basically a perfect demonstration of collaboration. An international collaborator who will bring a new angle to the project, a new expertise, a new aspect. Nowadays research, I've been involved in research many years, 25, 30 years ago, most articles, research articles were kind of monothematic. Nowadays, research articles have, they have biochemistry, molecular biology, structural biology, genomics, omics data. So you need to study the topic from lots of different angles. Usually, not always, but usually is the case. So use this collaboration that we look and also to the benefit of your project. The second thing is training. It's fundamental that young scientists are trained in the project. And again, here, they have to be young, you know, PhDs or postdocs, and names and surnames. You know, it doesn't have to be lots. It can be 2, 3, sometimes even, you know, oh, this PhD student will be trained on this postdoc will be, you know, and in many cases, oh, this PhD student will spend 3 months to our international collaborator, and these 3 months are going to be covered by the CRP. That's perfect. Also. So be very specific and be very clear in how collaboration and training will be part of your application. Okay. So all the application is all online. I'll go through, I almost finished the details, and we will ask the details of the PI, the title of the abstract. Again, you know, let me just say title and abstract are the first things that someone will read. So an overlong title, it doesn't work. You know, be nice and concise, both in title and abstract. You know, covering, you know, so by reading that, the reader already gets a very clear idea of where you're going, right? And then introduction obviously has to be a concise overview of the background. Again, here, if the introduction is not up to date, if the wrong references are cited, you then give the reader will then think, oh, does the applicant is really fully aware of the field? If you're citing the wrong references, or if it's not updated, if you don't know of the latest results, then obviously this will work against you. Okay. And the research project, you know, you will be asked about the aims, the milestones, the methodologies, the collaborative experiments, the facilities and training the facilities you have, the training that you will do, and the time frame. Again, as I mentioned before, everything has to be realistic and clearly stated. Okay, that I mentioned, the training I mentioned, you know, again, you know, I'm a bit repetitive because it's so important with respect to collaboration and training. Has to be as precise and as informative as possible. And again, in all spaces of the application online, do not exceed the space limit, okay? The CRP projects can go up to 36 months. They don't have to be 3 years, you know. Most, I have to be honest, most applications we received are for 3 years, but if the work you propose 2 years is enough, then put 2 years. Again, if we look at, you have to be realistic. Again, opposite of what I said before, if you ask for 3 years, then the amount of work that you propose, we see it doesn't justify 3 years, but can be done in 2 years, that will work against you. Okay. The maximum contribution is 25,000 euro per year. And again, you will see in our online application, the breakdown of this 25,000 euros. There's certain rules which are clearly described in our application procedure. For example, you can cover, you know, certain expenses like consumables, literature, some basic standard laboratory equipment, and travel and training costs. And all these voices have a maximum. So you cannot put, oh, I'm going to have 25,000 euros only of consumables. No, you need to have a logical breakdown, not exceeding the limits that are mentioned in our budget in our budget guidelines. Salaries of PIs of group leaders cannot be covered, and also we don't allow overheads, unfortunately. Okay. There's also a certain amount of funding you can use for external services like sequencing and so on. But again, even here, you know, there are limits of how you can use it. If for some reason, you know, you need to exceed by a certain, not too much percentage, for example, the external services cannot exceed, for example, 5,000 euros in your budget, but you need 7,000, then provide a justification. Or, you know, my project requires a lot of NGS, so I need to go to 7,000 and provide a justification. And this excess amount cannot be too much. Okay. Now, to finish, I have to mention the selection procedure because it will then reinforces everything I said, that really apply, and when you apply, it really has to be as perfect as possible. The first screening is organized by the liaison officer. So the liaison officer of Trinidad and Tobago, who you met today, you need to submit the grant proposal to her, and then the Trinidad and Tobago, like any ICGEB member country, can submit 3 standard CRP proposal in 2 early career maximum. If there are more, there will be an internal selection that the liaison officer will need to organize in order to select which 3 standard and which 2 early career can then be sent to ICGEB. A copy of all CRP applicants will also reach ICGEB, but the liaison officer will only endorse 3+2. But we have a copy of all of them here. Then once they reach to ICGEB, they will initially go through a triage procedure. So at least 2 ICGEB group leaders will read it, and they will do a first screening process. Now, let me tell you, I do this triage. This screening process is really looking whether the training is there, the collaboration is there, the timing is realistic, the budget is realistic. So everything I said, if collaboration is not there, or training is not there, or the timing is wrong, then it will be immediately discarded. If it fits, all the criteria I mentioned, if it's just, and then it will go to the next phase. So as I said, failure at this stage is written here is due mostly to improper presentation or lack of competence in people carrying out the work. And then, so if you pass this phase, it will go to peer review, and it will be sent out to 2 international experts. You have to appreciate this, because getting reviewers today is very difficult. The reviewers are, scientists are very busy in reviewing papers, grants. So we have to do a pre-screening, and we send it out to 2 international experts in the particular field of the grant proposal. Okay, so if you look at the typical numbers that we get, plus minus 5% every year is this. We have 500 to 600 applications which go to our liaison officers, and then we receive here in ICGEB endorsed 150 to 200. We undergo triage, as I mentioned. We look at which ones have a proper presentation, which have followed all the guidelines, and then usually 60 to 80. And these go out for peer review, as I mentioned. And usually 22 to 25 are funded. Our Director General is really fond of this program, is always trying to have more, but funding is always an issue. But usually 22 to 25 are funded every year. Okay. To finish, in our website, you will also find basically everything I said, probably if not more, how to write a grant proposal. It's written there, all the tips. Probably I missed something. You can clearly see it there. And also we have a Q&A session as well, file that you can download, questions and answers, which also hopefully will answer any questions that you have. If not, I put here. This is my last slide. Here we have specific emails for the CRP grant, the grant program, the fellowships program, the courses program, and also our Director General email, which if you write it there, then it can be your email then can be filtered through to the appropriate person who can answer your question. I hope I didn't take too long. I'm sorry for the chairman, but I wanted to make sure that I was clear. Okay, very happy to myself, Mariana, and Lawrence, and to answer any question you might have. Thank you.
Okay, thank you, Dr. Venturi, for that detailed explanation on the guidelines for applying to the programs offered by the ICGEB, including the coverage of the program and opportunities for personal self-expansion. Okay, so I would now introduce Mrs. Joanne Leong-Paul, Research Officer at NIHIS and ICGEB's Liaison Officer for Trinidad and Tobago, to give an overview of NIHIS's role. Under NIHIS's International Projects Unit, Ms. Leong-Paul has been involved in collaborating with international, regional, and local bodies to develop and promote joint matters relating to science and technology. These include projects such as Deep Sea Wonders of the Caribbean, Seismology in Schools, and a NIHIS NASA internship program. More recently, she has been involved in introduction and management of new initiatives relating to innovation and entrepreneurship at NIHIS's Fab Lab. Mrs. Leong-Paul, you now have the floor.
Thank you, Tahira, and good morning, ladies and gentlemen. I'm very happy to have you all here today and for us to be able to convene this session, because it really gives us some context into the opportunities that you will see being distributed yearly regarding ICGEB and our membership with them. So this morning, I'm just going to take you through a brief overview of NIHRS's role as liaison office for Trinidad and Tobago. Next slide, please. Okay, so here is the outline of the presentation. I'll touch on the background on NIHRS and our membership with ICGEB, the opportunities that we currently have available and our local process for applying. I'll give you a little brief on some of the evaluation criteria that we undergo in evaluating the research grants, and a snapshot of our distribution network who received the opportunities, as well as the benefits of our membership that we've received, as well as some of our success stories. Next slide. Okay, so a little bit about NIHRS. Right, so we are essentially established as a statutory body of the Republic of Trinidad and Tobago since 1984. And we are currently a state agency under the Ministry of Education. So our mandate is to promote the development of science, technology, and higher education in Trinidad and Tobago, and enhance the innovative, creative, and entrepreneurial capabilities of the general population. So looking at our mandate, we can see that there certainly is alignment with the role of ICGEB and our membership with them. Next slide. Okay, and just to provide some context as to our alignment to our strategic objectives and our strategic plan at NIHRS, the membership with ICGEB falls under strategic priority 1, which is science and innovation for development, and particularly strategic initiative of NIHRS research agenda, the goal of which is to deliver research results of today to meet STI challenges of tomorrow. I also like to touch on another of our strategic initiatives that I think our membership with ICGEB can also potentially align to, and this is our Technopreneurship Development Plan, where innovative research may lead to a patent potentially or improvement of TNT's global competitive index ranking. Next slide. Okay, so this is a snapshot of our history with ICGEB. Our previous speakers would have provided some of this information. Basically, back in the 1980s was when NIHRS worked closely with UE to spearhead the development of capability in biotechnology. Then in 2003, we officially became a member of ICGEB, with NIHRS being appointed the liaison office. In 2017, Cabinet appointed Professor Christine Carrington as our Board of Governor representative, following the retirement of the previous Board of Governor representative, Professor Julian Duncan, who some of you may know as the forefather of biotechnology in Trinidad and Tobago. And that brings us to present day, where our researchers in life sciences, and they now have access to all the ICGEB opportunities available through our membership with them. Next slide. Okay, so our previous speakers would have touched on this. I just put in this slide here to reinforce that for some persons who may think the area of biotechnology and genetic engineering to be, rather specific, ICGEB actually basically covers a broad area of topics under the whole area of life sciences. So next slide. Okay, and Dr. Venturi would have spoken about the 3 main opportunities in great detail that are available. So next slide, and I'll go into the local process here. So he would have also mentioned that the first stage of the screening would come from the local liaison office, where we would usually convene an expert of panels on the particular subject areas that have been submitted to us. Usually the panel would comprise 3 persons, and we would have them evaluate the proposal. What we usually do when we receive the opportunities from ICGEB is we then customize the artwork and distribute to the local network. And we ask for persons to submit 1 month prior to me the application documents, so that our panel can have an opportunity to go through and provide some guidance and feedback for applicants to perhaps edit as necessary before officially submitting through ICGEB's portal. So we do this for the meetings and courses and the research grants as the opportunities that require endorsement. So in that regard, I just want to highlight that, you know, it was strategic in hosting this workshop at this time to be ahead of the deadlines. However, the NEHRS deadline for the meetings and courses have passed, but we are going to extend it to ICGEB's deadline in order to encourage some applicants coming out of this session. And in that way, it will go straight through the portal and we will endorse based on what is there without having the opportunity to provide feedback for edits. Okay, and as Dr. Venturi would have also pointed out, you know, there are limited number of applications that we can endorse. So this is why, you know, we have this first stage screening process to ensure that we maximize and get the best out of the proposals that are being put forward. Okay, next slide. All right, so just a little bit on some of the things we look for when evaluating the research grants. We ask the question, if it's, what is the proposal addressing in life science in a concise and realistic manner? And to ensure that the timelines being proposed are also realistic. We also look for if the proposal is addressing a scientific pain point for the country. And then as Dr. Venturi would have also pointed out, we look for opportunity for collaboration and training of local scientists, as well as the necessary expertise required for carrying out the proposed research by the research team and the PI. Okay, next slide. Okay, and this is a glance of our database that we would usually distribute our opportunities to, because we do know that there's specific eligibility requirements. We tap into academia, research laboratories, and public sector and international bodies with national focal points who deal with research in life sciences. So at the end of my presentation, I have a link where if you wish to receive notifications on the opportunities directly, you can feel free to put in your information, as well as if you wish to propose any persons who we should add to our network, you can also use that link to do so. Next slide. Right, and just to end off, I wanted to give you a glance of some of the benefits that we have received through our membership with ICGEB. Over the years, we would have engaged in 2 technology transfer agreements. We would have received funding to co-organize 3 workshops, including funding of the participants, of 11 participants and 1 speaker, of 3 fellowship awards, 2 grants, and in total, a financial contribution of upward of 200,000 US. Okay, so next slide. And right, and I'll just take you through some of our success stories with hopes of encouraging more applications as we move forward. So in 2019, we would have had Dr. Varma Rambaran of UTT receive a CRP research grant to further his research on the synthesis and testing of novel drugs for diabetes mellitus. This actually builds on his work back in 2015, where he was also awarded a SMART fellowship to do work on alternative diabetes treatment. So Dr. Rambaran actually went on to file a patent in the US for his novel drug. I saw he was registered to attend, but I'm not seeing him here today. As I would like to hear, you know, some updates on what is the latest status with the patent. So also in 2018, we had a fellowship recipient, Dr. Emily Ramsahai, who did work on integrative biology. In 2016, we had Dr. Regini Haraksing receive an early career grant for research on genetic contributors to diabetes mellitus in Indo-Trinidadians. And her work was the first study of its kind in the country, and it serves as a landmark example for how genetic studies can be carried out in the country and the region, and the potential for their influence on the health of the Caribbean people. Okay, next slide. Right, and of course, we heard about Professor Carrington's award to host the 2015 VEME, and the impact it has had nationally on our local scientists and better preparing them for the role we took on during the COVID-19 pandemic. And our first recipient was back in 2005. That was Dr. Sefer Rampasad, who attended a fellowship to do research on virus-resistant transgenics. Okay, so that will take us to the end of my presentation. Here is the link for persons wishing to receive opportunities. I'll put the link in the chat, as well as this is also our website with our ICGEB page, our website link, where you could read more about our past success stories, as well as review yearly to see what are the opportunities ongoing. And my email contact, should you wish to reach out to me directly for any further guidance regarding any of these opportunities. Okay, so that brings me to the end of my presentation. Feel free, I encourage you to put your questions in the chat as we go, because we do have 2 other presentations following my session. And at the end, we have a forum for open discussion. Okay, thank you.
Okay, thank you, Ms. Leong-Pao, for that informative presentation on NIHIL's role and sharing the evaluation criteria and showing some of our past applicants. And now I would hand it back over to Dr. Vittorio Venturi to give us a brief overview on biomedical research, followed by a brief overview of agricultural and environmental research. So we had a little change in the agenda where we have Dr. Vittorio Venturi given the brief overview on biomedical research to replace Dr. Banks. Okay, thanks. Thank you.
Thank you. Can you hear me and can you see my screen?
Yes, yes, I can.
Okay. So yeah, I will try to be briefer because I talked a bit long before, because I thought it was particularly important. I will be a little briefer with respect to looking at, so as Tahira mentioned, I will discuss both biomedical briefly and environmental research in ICGEB. But again, I will try to be brief. But again, I draw your attention to our website, which is constantly updated and will as informative of all the names and surnames of our PIs and the current research topic, and also you will see latest research highlights with latest publications. So really my suggestion is to go to our website. So very briefly, the research pillars at ICGEB that we in the life sciences that we cover can be also, as Mariana mentioned, like these can also be called macro areas. We have several scientists working on cancer, different aspects of cancer I briefly mentioned. We have groups studying infectious diseases. These are mainly, there is a predominance of virologists in ICGEB, but also scientists looking at malaria as well as tuberculosis. We have a scientist looking at other non-communicable diseases like cardiovascular diseases or neurodegenerative diseases. And we have also scientists in plant biotechnology, as well as some industrial biotechnology, especially biofuels. Let me just say here that there are group leaders in all three components that are doing research in these macro areas. It is true to say that in Trieste, we have a predominance of scientists looking at non-communicable diseases, so non-infectious diseases, so mainly cardiovascular, cancer, and neurodegeneration. And most of the plant biology and biotechnology groups are located in New Delhi, okay? But again, there will be also groups in Trieste and New Delhi and Cape Town, which are involved in the other pillars as well. So I don't want to spend time here. Already you've seen from Mariana, you will see on our website the macro areas with all the names and surnames of all the group leaders that are involved in that particular macro area. Okay, so when we look at biomedical research, we have communicable diseases. So in virology, we have scientists looking at dengue, chikungunya, and COVID-19 mainly, but we also have some other flavivirus and arboviruses that are being studied. And a lot of the research is going on is some epidemiology studies, as well as mechanistic studies involving entry of these viruses, and also looking for antiviral strategies or drugs to block their infection. With parasitic diseases, we have small worms, helminth studies working with malaria, and again here we look at looking some mechanisms of host pathogen interaction, as well as looking for anti-parasitic drugs. So, and also I must say that some groups in New Delhi are very translational, so they're very looking at applications, and maybe in Trieste looking more at fundamental mechanisms. Okay. With respect to non-communicable diseases, I will mention some slides of some groups looking at cardiovascular disorders. We have groups mainly in Trieste in immunology, in groups in New Delhi and in Trieste looking at response to pathogens and cancer. In neurobiology, we have scientists in Trieste, and I will mention briefly. And also in cancer, we have several scientists who are very rich in scientists studying cancer. Can be through how the human papilloma viruses infects and generates cancer, and also studying cancer microenvironment, the cancer microenvironment, and also cancer genomics. And recently we have invested in computational biology for obvious reasons. Now we are there's a lot of omics, let it be transcriptomics, genomics, metabolomics, or proteomics that are being done. So there's a lot of data generation at the moment in science. So the ability to analyze this data efficiently, not only the data that you might generate or we generate, but also integrate the data that is available out there in databases is becoming very important. So very quickly, again, I don't want to take too much of the time, but for example, cardiovascular biology here in Trieste, we have a group leader that is really beginning to be interested in studying why there's a low incidence of cancer in the heart. Is that also linked to the fact that the heart cannot regenerate itself? So looking also how we can improve lung regeneration through the role of endothelial cells. So the two topics are a little bit connected, looking why cancer is not being developed and how we can regenerate the heart through endothelial cells. And similarly, another group here in Trieste has looked and is looking how these microRNAs, so microRNAs are present basically in every living organism, also in plants, how these microRNAs can be used to stimulate cardiac repair or cardiac regeneration. For example, after myocardial infarction, they've done high throughput screens. They've had very highly successful results because they've isolated a few microRNAs that can, when applied, they are very effective in heart regeneration or cardiac repair. And now these microRNAs are also taken further to see if they can be used as potential drugs to cure this condition. Okay. Again, also related to COVID, I'm not going to talk too much about COVID, but at the COVID outbreak, our scientists mobilized in all our three components and have been involved, and some are still involved in COVID-19 research. And one of the discoveries that we have made is that scientists here in Trieste have shown that during COVID infection, the pneumocytes of the lungs actually undergo form syncytia. So basically cells fuse and form very large cells which have lots of nuclei. And then once they've discovered this, they've also then done a screening using a high throughput facility that we have here in Trieste, which I'll briefly mention, using FDA approved drugs. And they found a drug that was able to inhibit the production of these syncytia, so this fusion of cells, which was actually stimulated by the presence of COVID-19 infection of the pneumocytes. So and this drug is an FDA approved drug, so now it's under test if we can be used to cure or at least in some way prevent COVID-19 lung damage. Okay. So we also have immunology groups, and again here, especially we using the latest techniques. For example, here a group in Trieste has set up conditions to identify and isolate a rare population of immune cells from tumoral tissue, and these they use multicolor flow cytometry. So so they can separate these cells, and then hopefully these cells and studying the mechanisms can then be useful and helpful in developing immunotherapy, which at the moment is very exciting. Okay. The HPV group actually of Lawrence Banks here in Trieste has been working on human papillomavirus for many years. And they're looking how, for example, they're using currently using latest technology in genome editing, demonstrating our protein, the E7 protein, can actually, by blocking phosphorylation of this protein, can be a valid therapeutic approach because it really stops the HPV oncogenic induced malignancy that it causes. So again, this is a very good cancer research that we've performed in Trieste, and we also perform in Trieste. We look at the microenvironment of cancer. Another group is looking at the microenvironment of cancer and how this microenvironment is controlled and how we can, how cell-to-cell signaling takes place in generating, let's say, this microenvironment. We also have groups in neurodegeneration, and in particular, we have a group looking at TDP3, this protein. When it accumulates, it causes neurodegeneration in several disorders, and in particular, this group is looking at ribonuclear proteins, which are basically complexes of RNA and proteins, and how these RNA protein complexes affect the splicing mechanism of this TDP43 locus, which then results in hyperaccumulation of the protein, which then results in neurodegeneration of several neurodegeneration disorders. Okay. Importantly, here in Trieste, also we have a high throughput screening facility that is actually run by one of our group leaders, and this allows very robotic screening, fast screening. So robotic screening together with a high throughput microscope for screening for any kind of activity within a cell. You know, if you have, let's say, You can screen, especially using autofluorescent proteins or cell size. So different parameters of cells can be screened by high throughput using this robotic and high throughput microscope. So we can screen, for example, tens of thousands of different effects of different, for example, microRNAs or RNAis or drugs. When you have a particular assay, this can be screened very efficiently, very quickly. So again, this is a group which provides a facility which is available to all our ICGEB laboratories from ICGEB member countries. So if you think that you have an assay that you want to screen for a drug, or you want to screen or you want to screen you have an assay that you want to screen something, you should contact this group and initiate a discussion whether you can set up a screen here in Trieste. We have here we also have biotechnology development units both in Trieste and in New Delhi. In Trieste, they're very much focused on biosimilars. Okay, so we have a new pharma compliant laboratory for biosimilars. So we have a portfolio of almost 15 proteins, biosimilar proteins so we have the process to produce them, and we also have a pilot plant to produce them all under GMP conditions, and also to do QC once they are produced. So we transfer this technology to anyone who is interested, mainly companies in our member countries. So they come here, they learn the pilot process on how to use the biological system to produce it. It can be a eukaryotic cell line, it can be a fungus, or it can be a bacteria, depending on the protein that is being produced. You learn how to produce it under GMP conditions. You then purify it and QC test it. Okay, and then once you do this training, you can also leave with the biological materials in order to then clone this procedure back home in your company, and then scale it up and then reach the market. Okay, so we have all this facility here in Trieste, and it's all in the website for you to read. This is very quickly a list of the technologies, and you can see here we use all different chassis depending on the product. For example, for E. coli, we produce, for insulin, we produce it in E. coli, and then we have interferon, we have erythropoietin, and so on and so forth. What is important to see here, that the cost of production is very low, much lower than the cost of if you go to the chemist and buy these drugs. So a country can produce these drugs at very low cost and make them available at a cheap cost to their population. And we've done, as Mariana said, we've done a lot of these transfers. We've done over 90 of these transfers in our ICGEB member countries. So again, I don't want to mention too much on COVID, but the COVID mobilization was incredible in ICGEB. We have several groups that repurposed in COVID research, and these are all described on our website. What I want to mention is that we have also a special project with the Bill and Melinda Gates Foundation in order to bring cheap COVID tests and COVID monitoring, especially in Africa. So we have special programs in COVID, again, all described on our website, and also researchers looking for anti-COVID drugs, and also mechanisms that COVID uses for entry into eukaryotic cells. Shifting to plant biology and biotechnology, we have several groups that are looking at, I've put here some of the topics that we look at. Plant transformation technology, we use the latest techniques, and then we do engineered herbicide resistance, crop improvement via genome editing, improve micronutrients in plants, plant pathogen interactions, looking at also microRNAs. I mentioned microRNAs that can be used for example, which can be useful for heart regeneration, but also microRNAs are very important also in plant development, in plant life. So they're involved in many different biological processes in plants. And apart from biotic stress, we'll also look at abiotic stress, very important, especially in this period of climate change. We're looking at salinity, especially salinity and drought resistance, okay? And we also initiated a program recently on plant microbiomes. Thanks to NGS, we now aware, we knew before, but we are now aware that just like humans, plants live together with a lot of microbes. So how do these, what kind of effect these microbes have, what kind of interactions they have with the host, with the plant host, and how can they be useful in order to develop sustainable ways for plant growth, and by reducing the use of agrochemicals. So very briefly, for example, the group in India is actually used genome editing to generate glyphosate-resistant crops. Glyphosate is an important herbicide which is used in farming, extensively used because it kills all the weeds. Whereas the crop that we want to grow, we have to make it glyphosate resistant, and we've done so. We've done so very effectively so far using transgenic plants that carry a bacterial gene. Glyphosate attacks an enzyme that is involved in amino acid biosynthesis, and the bacterial gene is resistant to glyphosate. Now, what scientists in New Delhi have done, they have taken the endogenous gene that is important for this amino acid biosynthesis, and they changed a few amino acids and have made it glyphosate tolerant. And they've been able to do this via genome editing. So using latest genetic engineering technology, by working on the endogenous gene, we can then make very important changes in plants. Similarly, in New Delhi, looking at plant pathogen interaction. Again, we have groups, we have also more than one group that does this. But what I want to say here, for example, in this particular slide, this group is looking at how the brown plant hopper is affecting rice growth in India. But what is important is that this group is using all the latest technologies and techniques in order to study this plant pathogen interaction. For example, is doing microbiome studies to see if the microbiome of the plant hopper plays a role in making the plant hopper effective and pathogenic. Okay, they're doing also population genetics of the plant hopper to see whether there's some lineages or some genotypes which are more important, which are more virulent than others. They're doing RNA transcriptomics through RNA sequencing to try to understand which genes and which systems are important for the plant pathogen interaction. So they're doing transcriptomics in plants and also in the insect. And also they're doing all other kinds of interactomics, especially trying to understand which proteins are injected into the plant cells, which can of the insect, which can then be, if counteracted by the plant becomes resistant. So looking at identifying resistant proteins of rice varieties, which can then be used to create rice which is resistant to this insect. So again, you see that we're using all the latest techniques to study interaction between a pathogen and a host. And also these techniques are not only used in plant biology, but they're also used in looking at parasites or virus in the biomedical sector. Okay, so again, what I mentioned earlier, if any of you wants to spend, for example, a short-term visit and looking at one of these techniques in order to study your plant pathogen model, let it be in biomedicine or in plant biology, we have the expertise to help you in this, or to collaborate with you in this particular technique, novel technique, in order to study plant pathogen interaction. Importantly, we have a unit, a big unit in New Delhi that looks at biofuels. So there are several group leaders that are looking at different aspects of biofuels, let it be mainly second generation, third generation, and even fourth generation biofuels. So, for example, in this slide, they've done many years research to identify enzymes produced by fungi that are important for the pre-treatment of agricultural waste. For example, once you harvest your rice or your corn or your wheat, the waste that is left is obviously, they are carbohydrates, but they're all polymeric carbohydrates, which are not free freely usable, for example, for yeast, for fermentation. So scientists have worked very hard to generate cocktails of high amounts of cocktails of enzyme by identifying firstly the fungi, then engineering the fungi to make large amounts of these enzymes. So then this enzyme extract can then be applied to this agricultural waste. So these polymers, these sugar polymers can then be digested, and simple sugars or free sugars will be released. So then they can be used by, for example, yeast to generate ethanol. So you can generate bioethanol from agricultural waste. So it's been very successful in this research. And also there are scientists looking at third generation biofuels, basically isolating microalgae or trying to understand how we can improve fat accumulation inside microalgae, which can then be purified and converted into biodiesel. And then also through fourth generation biofuels, doing genetic engineering, having microorganisms producing long chain alkanes. Finally, as I mentioned, we're very interested in plant microbiomes. So we now have research in all the three components. We initiate, through a collaboration, trying to understand plant microbiomes, especially in crops like rice and sorghum, understanding what kind of microbiomes they have, which are the major actors in the microbiomes, and then trying to isolate these microbes and see if we can utilize them either to improve plant growth as a biofertilizer, or to allow the plant to be more resistant to pathogens, because these microbes allow the plant to keep the pathogens away, either directly by killing the pathogen or indirectly by inducing the immune system of the plant. So we have, for example, done experiments. We've compared, for example, diseased plants versus healthy plants, and then we looked at the microbiomes of both, compare these microbiomes, and then that will give us an idea which microbes are important for keeping the plant healthy. And then these can be isolated and studied in the greenhouses and in the field to see if we can use them as probiotics or as biocontrol agents. We also are very interested to promote the use of, for example, biopesticides, but in general of bioinoculants, and as these can be very helpful, especially in Africa, in overcoming barriers of trade, because some agricultural products coming from some countries in our community have too high doses of agrochemicals. So by using biopesticides, we reduce the use of agrochemicals, or having an integrated approach using agrochemicals and biopesticides will reduce the use of agrochemicals. So then the final seed or the final product that we want to sell has below alarming levels of agrochemicals, so they can be exported worldwide. And in the moment, there are some countries that produce some agricultural products which have too much agrochemicals. So we support very much the use of biopesticides. And through networking, especially in the South African developing community, in the SADC community, we have a special project in order to promote this and to promote, which will result in more trade. And we are looking whether we can do this also in many other countries in our community, promoting the use of much more sustainable ways to do agriculture in the future. Okay, so I finished there. I just mentioned that, again, in our website, you can also find a lot of facilities that we have. I mentioned some of them here. I mentioned, in this brief presentation, I mentioned already high throughput sequencing, but we have also other facilities, proteomics, flow cytometry, microscopy. We also make, as I said, I mentioned the BDU, the biotechnology development unit, to make biosimilars. So we have several quality control techniques. So again, go to our website, and if any of these techniques is particularly useful or absolutely necessary for your project that you're doing and is not available in Trinidad and Tobago, again, this is an excellent way to apply for a short-term fellowship, or even just contact the group. Sometimes it's even possible to make collaborations by sending samples or sending sending biologicals, and then a collaboration can also be initiated from a distance. Okay, I finished, and that's my email in case you wanna contact me. Again, I apologize for being brief, but I believe that these remote Zoom meetings should not exceed a certain amount of time, because I've seen in the past that after a while people tend to the concentration level tends to decrease. Okay, thank you very much.
Okay, thank you, Dr. Venturi, for providing some insight into some very dynamic topics around biomedical, agricultural, and environmental research. All right, so we now welcome any questions as we move on to the end of today's workshop. The floor is now open for discussion.
Looks like everybody's tired. We actually had quite a lot of exchanges in the chat, so I think that that maybe a lot of the questions were dealt with as they came up.
So are any of the students who attended interested in pursuing some applications? Maybe they're a bit shy.
We will see after the deadline what comes in.
Okay.
Sure, you will get questions after and interest.
And Christine, I guess this will be, this has been recorded, so it can be made available.
Yes.
Maybe even on your website or whatever, for anyone who wants to have a look at it and get more information, even if they couldn't make the workshop today.
Certainly.
Okay.
Shall we move on?
Okay, so no questions. All right. Well, thanks everyone for. Well, if you do have any further questions, please feel free to send it to us in an email, and we will try our best to get them answered. I now invite Professor Christine Carrington and Dr. Lawrence Banks to give the closing remarks alongside each other.
Yeah, thank you very much, Tahira. But I think today we heard about all the ways the ICGEB supports countries and researchers in sort of harnessing the power of biotechnology and molecular biology towards capacity building, whether it's national, regional, international capacity building and development through their grants, their short and long-term fellowships, and support for workshops and courses. And I think we got a lot of important information on the ICGEB's mission, its governance activities, its program initiatives, and also the role of NIHURST as the liaison institution. I was particularly pleased to hear about the fact that the collaborations that the ICGEB encourages go beyond the established centers of excellence in Italy, India, and South Africa. And I think that's an important reminder. There's 64 other ICGEB member states out there. So there's a lot of scope for collaboration and networking and cooperation. And Dr. Venturi gave us detailed guidance on the funding eligibility and how to produce competitive applications for each of the different types of programs, and the comprehensive overview. Although he said it was short, it was quite good, very good. Good overview of all the research done at the ICGEB centers. And I hope that everyone who saw it found something that's synced with their own research interests, and I encourage them to, reach out to the PIs about collaboration. And I'd also like to encourage everyone who participated to share what they learned with their colleagues. Some of us are too old to access some of the initiatives, but we all know young scientists who are home or abroad who can benefit. So I hope that you spread the word. And as we heard, the ICGEB has a commitment to open science. So all of this material, as we heard, is online, or from today's workshop is going to be online, and all their previous workshops and lectures are available to us. So I encourage people to access them, share them, and learn from them. And I am confident that what we've done today will increase the number of individuals who are aware, and also to improve the quality of our submissions, and I look forward to seeing them. And I just think for me, just to say thank you for those who joined us, and thank you to my colleagues on the panel from the ICGEB and the NEHIS for their participation today.